Bueno, sean muy bienvenidos. Es un honor para nosotros contar con vuestra presencia. Voy a presentar a Nora. Ella es editora del diario La Nación en Argentina, es especialista en el tema de la ciencia y ella será la encargada de entrevistar al doctor Sierk Poetting. Me gustaría que él se presente a sí mismo, así que, Dr. Poetting, would you be so kind to introduce yourself to our audience, please?
Yes, thank you very much for the kind introduction. Yeah, I'm Sierk, me llamo Sierk, Sierk Poetting, and I'm the CFO and the COO of BioNTech. So I joined the company 7 years ago when the company was still small, around 300 employees. And I'm a physicist by training. So I have a PhD in quantum optics. And after quantum optics, I thought I have to do something real life and implement things. So I went to did some consulting actually at McKinsey for 2.5 years basically, and after that I came to Sandoz, so Novartis Sandoz. So I worked as a consultant in the integration office when there was a big merger in 2005, and then I stayed at Sandoz, so basically Novartis for 8 years. So I was in the generics division, in the Sandoz division for 4 years in the headquarters and 4 years in the United States. And after that, I joined BioNTech, where became CFO, COO in 2014. And since then, I've been there, and the company changed quite a bit in the last 7 years. So I'm happy to talk about this in a few minutes.
Thank you so much, Doctor. It's an honor, and we know it's a special treat. This wasn't in our original program, so we are thrilled to have you with us. And I understand that you will be sharing a presentation with us. Is that correct?
That is correct. That is correct.
Wonderful. So I will ask our director, Gonzalo, who is in Spain at the moment, if he would please introduce and allow you. The audience is all yours. Thank you so much.
Very good. So let me start. So today I would like to talk about and show you more about BioNTech and the future of immunotherapy. So basically, what does it mean? So we are talking about where did the company come from, because this is important to say why were we able to develop and produce the first vaccine against COVID-19? Why were we ready, and what else are we doing actually? So why can we produce this stuff? What else are we doing? And this is a little bit what I want to talk about. And the talk will be around about 30 minutes, and then I think together with Nora, there will be a question and answer session, on anything you want to know basically. So let's start. So a little bit of like disclaimers and stuff, because we are publicly listed by now. But now here comes the story. So BioNTech was built and founded in 2008. So basically we are now 13 years old, and it was founded as a next generation immunotherapy company. So why immunotherapy? Because if you can harness the immune system, if you can program the immune system, you have a perfect weapon actually to go against infectious diseases as a vaccine mechanism, but you can also use the immune system to clear up cancers in the body, so really fight cancer. We'll go into more details why we can do this and how we do this, but this was the idea actually, harness the immune system to do this. And but how exactly do we want to do this? So first of all, we wanted to really build an integrated biopharmaceutical company that does novel things, disruptive technologies. So not me too's or small alterations of stuff that's there. That's important. I mean, generics industry is very important. I've been working there for 8 years, but this company was founded to do new stuff that hasn't been existent, or that was like far-fetched, or more or less like a moonshot. So, and that meant you needed a fully integrated company. First of all, the dream was, or the company set off to have own products on the market. So this became true now with the COVID vaccine. We thought this would take a little bit longer, with the first cancer vaccines coming probably down around about like 3 years or so from now. This is probably when we anticipate the first cancer products in the market. But the company was really founded to be like a broad company with own marketing and sales, with own production, and with a huge research organization. And as I said, own products on the market, not like a one-trick pony, do something, get sold to some other company, but really be there as a company. And for that, you need a lot of things. You don't only need like research and development, but you also need, like, as I said, manufacturing. So as a small biotech company in 2009, 1 year after foundation, we bought already a GMP facility to be able to produce everything that we are doing. Why? because most of the things are so novel, we weren't able to go anywhere into contract manufacturing organizations or so. We had to do it ourselves. And plus, you have the opportunity then, if you have your own manufacturing, you have like very iterative and small innovation cycles. You can tech transfer it to your own facility, and it's basically there whenever you need it. You don't have to, reserve slots and stuff like that. That's why we had like GMP manufacturing from the beginning onwards. And we also, because if you want to leverage the immune system, you need to know what you're looking at. You need to identify the cancers. You need to identify the virus and the immune responses to the virus and stuff like that. That's why we also have an own diagnostics unit, actually. So we set off with this, like, let's call it a broad suite of technologies, a fully integrated company, and wanted to develop immunotherapies against cancer and infectious diseases and beyond. And I'll explain you what the beyond could be in the end of the story as well. And what you also need is you need partnerships and global collaborations. And let me quickly talk about this for a second, because the way the company was founded was actually fantastic. It's a lesson how you do this. So we had our founders, Ugo Sahin and Özlem Türeci, whom you know. They are a couple. They've been working. They're both physicians and oncologists, and they were frustrated because there was no cancer therapy around. So they basically said, like 25 years ago, then we have to do it ourselves. So they set off. They said the research. They had a first company that was founded in 2001, and then another company, the BioNTech, founded in 2008. So they had a vast strategic idea, 20, 30 years of pipeline in their head already, and they started to build it, and they needed financing for this. And at that point, the Stringman family came into the play. The Stringman family, having built Hexal, sold it to Novartis, and then invested basically $1 billion into biotech. They invested 150 million together with a smaller fund in the beginning into BioNTech. So there was this long-term investor with the Stringman family office, and the couple with the ideas of like innovative ideas, and both together said like, we want to build this company. And that's why they set out actually with 150 million euros, biggest seed funding round in Europe at the time in biotech ever, set out with this like big amount of money and the big bag of ideas into the future to create technologies against infectious diseases and cancers. And it's very important that it's many technologies. It's not only the messenger RNA technology that we're going to talk about, but there's other stuff as well, because in the end, it doesn't matter how you hit your enemy, be it cancer or infectious diseases, you have to have a good technology that suits. And for the COVID vaccine, it was mRNA that fit best. So looking under the hood a little bit, what do we have? So we have a deep expertise in immunology. That's what I mentioned before. So both our founders came from the medical side as doctors. So, and they really learned the immune system. They understood the mechanisms, how you can prime and influence the immune system. So then they built cutting edge platforms for a couple of years. So basically we didn't go outside, we didn't have a web page, but in stealth mode, they were able to actually work on these 4 platforms, a small molecule platform and a big mRNA platform, a cell and gene platform, and a virus-like particle platform. So basically all of this together, this was built and iterated and optimized, and a couple of years later, when we started to go out and partner, that's when the platforms were actually at a level of maturity where you could show to other big pharma companies. So platforms, deep immunology expertise, and of course, then came bioinformatics. So everything that we do, we simulate, we learn, we drive with artificial intelligence and machine learning. We drive our conclusions from the data that we have. And as I mentioned before, we have in-house GMP manufacturing capacity for mRNA, but also cell and gene therapies. So that all of this together, we had when the COVID virus came actually, and we were ready to approach it. So now, how do we develop now? So we do have this vaccine on the market now. We proved that one of the platforms, the mRNA platform, is actually really working. So this is a breakthrough because now we can use all the other tools, and everybody believes us that mRNA is really a powerful tool. So with that, we have Comirnaty, which is the brand name of our vaccine together with Pfizer. So now we have this in the market, and with the cash flows from from selling that vaccine, we can accelerate our broad portfolio. Actually, we have more than 20 portfolio pipeline products that are ongoing in various stages of maturity, actually, and some in phase 2, many in phase 1, and a very big bunch actually also in preclinical still to awaiting clinical studies. So we can now drive this massively and in parallel through the clinic. And if this works, and if the mRNA technology works, and we have lots of science that this also works in messenger RNA therapy, but we also have like bispecific antibodies and cell and gene therapies and stuff like that. So if this really works, we might actually usher in a new era of vaccines and immunotherapies. So what we really want to build is like a multi-product immunotherapy powerhouse. So how did we do this? So this is the game, this is the end game, this is the goal. So how did we get there? First of all, when the 150 million of the seed funding ran out, and this is about the time when I came to the company in 2014, all the platforms were ripe actually to be partnered. So we didn't want to sell a lot because we really believe in the platforms and we believe in what we are doing. So most of the deals that we did were 50-50 deals, i.e. Cost sharing and profit sharing deals. So basically we said like, you have to pay a little bit of like, we need some financing from you guys from Pfizer or Regeneron or Genentech or Genmab and Sanofi and stuff like that. So we need some financing from you guys to get you onto our platform. So there were upfront payments and equity investments done. But then we said like, we want to develop it together with you, not only because we needed partners. We are not able to do really large scale clinical trials yet, but we, for example, brought the clinical manufacturing of all these stuff, and we brought the science to the table. And the other partners, for example, ran like large scale clinical trials, for example, in the COVID trial now and stuff like that. And they brought expertise and so on. And that's why we said we need to do that as a partnership, and we want to invest into this. That's why we said like, let's halve the cost. And if there are profits, let's have the profits. So most of our deals are actually this like half business model. What we also said, because we eventually want to be on the market and have our own products, we also have co-commercialization rights. So we said like, maybe now in the case of Pfizer, you can do many countries in the world, but we want to do a couple of countries to get up and running on commercialization. So when our big cancer products actually hit the market, later in a couple of years, hopefully, then we are able to also sell the products ourselves. So it's really, really commercialization with training wheels, so to say. So all of this was built into the deals already, again, with the end game in mind that we want to be a fully sufficient, fully self-maintained company. So these are the partnerships that we went into in various stages of the maturity. It helped us to bridge the years between 2014, when basically the money ran out this initial seed funding, and 2019, when we actually went public, and then we had access to the stock market. We did some other Series A and B funding in between, but those were of course mostly non-dilutive deal fundings here with these partners, and that helped the balance sheet and helped also bring our programs forward by having really excellent partners here. So now all of this accumulates into the timeline for the vaccine. So how did this all start? So basically we were sitting there in 2020, came out of an IPO, we just went public at NASDAQ, and we thought our balance sheet was not very rich at that time. And we thought like, okay, we have to have like a consolidation year. We hired many people, and we said like, everybody has to find their place. We have to refocus on our cancer programs, and then really push through, do some additional financing, and then push some cancer products, get some cancer products really accelerated into the clinic. That was the goal in beginning of January 2020. End of 2020, January 2020, it became apparent to Ugur and Özlem that this will be a pandemic. So that the virus from China, from Wuhan, that this is actually something that will probably end in a pandemic. So in late January, we started already with research and development on the vaccine. So we had like 20, 30 mRNA candidates tested already because we had the technology, and we started initiating preclinical data collection already in early February. So we went into the lockdown very quickly, and then we did everything at the same time from February onwards. So we had to look for financing because this was not planned in the budget. So actually we went ahead and and started a discussion with Fosun. Fosun also had like data from China for the virus. They were a really good collaborative partner in the development. So basically, the Fosun deal was to have a partnership on the development, but they also contributed some money. So that money enabled us to buy raw materials and stuff like that fairly early on in the process already. Because if you have money and if you can develop a vaccine, this only works if you also can produce it. Because if you cannot produce it, it's worth nothing. So we started at the same time in March already to buy all these raw materials and set up a new production network. So what this means, production network, the mRNA that we were able to produce at our site was in the 1 gram scale, 1 gram per week or something like that. That's what we did at most in our facilities. And now we are talking several kilograms per week, actually. So this is a factor of like 500 to 1,000, how we had to scale it up to answer to a pandemic. So we really had this like three-pronged approach, three actions in parallel. We had to look for financing, we had to develop the product and get it into the clinic, and we had to ramp up the manufacturing network. So Fosun was the first step. The second step was that we got Pfizer on board as the partner for non-China or non-Asia. That was very important because they also contributed money. We also had a profit and cost sharing, but they also added vast capacities because it is a scaling monster. Let's put it like this. They can run big trials and they can really fantastically scale up manufacturing. So it had to be a partnership. And all of this together enabled us to start a phase 1/2 trial in April already. Last week of April, we started our phase 1/2 trial. And with the data readout that we got from this in summer, we went on to the FDA Fast Track designation already in July. We initiated the phase 2/3 trial actually in July, end of July. And from then on, we initiated rolling submissions. We submitted at the EMA, to Canada health authorities, UK health authorities, Singapore, New Zealand, stuff like that. And we finally got the data, the phase 3 data after 45,000 study subjects being dosed with a vaccine efficacy of 95% in November. We handed in all the data in this rolling process. And then in December, we got the first approvals. And by now we are in more than 70, actually by now it's almost 90 countries that we're worldwide rolling out our vaccine. And we are closing to the 900 million doses shipped mark this month, actually. So this was a very compressed development timeline with like ramping it up, doing a very big scale trial and financing the company. So this is how we did this. And now, how did we do it? So why is messenger RNA so well suited for these things? So messenger RNA, and this is, I think, as much scientific as it gets today. So the great thing about messenger RNA is you can really program the immune system. So how do you do this? So we have been working on mRNA. I mean, it's only one of our platforms. So we have other platforms, but this is the most mature platform we've been working on the longest. I mean, we've been working on mRNA for more than 20 years now. And we have a couple of versions of mRNA. There's immunosuppressive mRNA, there's immunostimulating mRNA, there's a self-replicating mRNA, etc., etc. And we have like all these things, like the cap is optimized, and there's this 5' UTR, there's the 3' UTR, the poly 8 tail. All of these pieces of the mRNA are actually optimized, and we are able to pick out of our toolbox. We actually tested clinically a couple of these, and we found that one of these structures was the best one. Actually, with that, we went into the phase 2/3 trial. So mRNA is a natural molecule, actually, which copies data from the cell, from the DNA. It copies data, one gene, and it transports it. So it basically contains information, i.e. the message, that's why it's messenger RNA, from the core of the cell into the ribosomes, the factories, so to say, in the cell. So basically this one gene, the information of this one gene is transported to the ribosome. In the ribosome, a protein is built from the blueprint that's encoded on the mRNA. So basically the mRNA, it contains information. And with that information, the factories in the cell can produce whatever you would like to produce, have produced by the cell. So this is the powerful concept. Now, if you don't have natural mRNA, the mRNA gets degraded very quickly by enzymes and stuff like that in the body. So that's why we had to optimize all these things with cap structures and the blocker and linker structures and so on, so that it's not degraded. We also worked heavily and in-licensed a lot and did our own development actually on this like packaging here. So you package the mRNA in lipid nanoparticles so that it goes to the cells that you want it to go to, and that it doesn't degrade before that. So having all of this optimized, now we can actually insert RNA into the cell, and then the cell produces the proteins that we wanted to produce. So in this case of the COVID vaccine, we basically have like the structure ongoing, the production process is the same for everything. We just introduce the information of this protein that we want to produce. And in this case, we produce the spike protein. The spike protein is the little crowns on the COVID virus. So basically, we encode this. The cell actually produces those, and then if you find the right cells in the body that prime the immune system, these are produced, and these so-called dendritic cells then actually program your killer army. For example, they produce or they show these the spike protein to CD4 plus helpers T cells. And CD8 plus cytotoxic T cells, so basically killer cells. It basically tells the killer cells, if you see something that contains the spike, i.e. this little crown, eat it up. So infected cells can be eaten up by the T cells. And on top of that, the B cells are programmed with this like information of the spike protein. They produce neutralizing antibodies against this. So then you have like active T cells and antibodies because you showed the body the spike protein here. And that's why it's so great, a little bit of forward, if there is now a new variant of concern that is really bad actually that we need to address, the only thing that we need to change is the information here. Instead of the spike protein, we have to have a very mutated spike protein in here, and this stuff will probably very likely work as well. So this is the mechanism, and that's why it works so greatly, and because of the optimization for many years, this is so effective. So if you think about this now, what we showed now, mRNA today, first mRNA product on the market, Comirnaty, our COVID-19 vaccine together with Pfizer on the market, worked great. But this is now a new drug class because nobody has done mRNA before. So what do we do next? We can work on other infectious diseases with the mRNA. We can work on cancer vaccines. We have been working on this actually. This has been the focus of our company actually since inception. So we have like very advanced clinical trials and lots of data on these cancer vaccines already, including cancer vaccines that are produced just for one patient, so fully individualized. We have CAR T cell programs where you can amplify the CAR T cells by mRNA. And we have like systemic mRNA encoded immunotherapies, ribocytokines, for example, that are encoded, or antibodies that are produced based on mRNA that's moved into the body. So all of this is coming tomorrow. And then beyond that, so this is in the pipeline already. And then beyond that, beyond the horizon, you can work with mRNA on autoimmune diseases, rare diseases, and a lot of other therapeutic areas actually. So if you just go into the piece of like the infectious diseases, so there hasn't been very many infectious disease vaccines approved in the last couple of years, only 7 in 3 years actually. And there are so many where you don't have anything approved yet. So and the success of COVID actually shows us that mRNA can probably tackle stuff that hasn't been possible before. So Comirnaty, we did this. So now there will be the next generation COVID-19 vaccines. So for example, against new variants and stuff like that. This can be done. Then we are working on influenza, we are working on HIV and TB, tuberculosis with the Melinda Gates Foundation. Influenza is another project with Pfizer together. And then we have like 6 undisclosed projects we are working on already right now. So now, having developed this and working on this is great, but now again, you have to produce that stuff. So by now we said together with Pfizer, we are targeting 3 billion doses to be produced in 2021. And probably we have like more capacity in 2022. In this global network together with Pfizer, more of 50% of the drug substance is actually coming from BioNTech. Whereas Pfizer, for example, does a lot of fill and finish and a little bit more on the drug product side, on the formulation side. The Marburg site that we got online, this is the big site in Germany that we bought last September actually, or in October we were able to move in. That we got up and running, delivered the first batch already. We are ramping up to a three shift, full shift, 24/7 shift model. And by end of the year at full capacity, this will have 1 billion doses capacity that we brought up online very quickly actually. And with that, it's actually the largest mRNA, one of the largest mRNA production sites worldwide. And when you look at what has to be done, I mean, it doesn't look that complicated, but it's all novel in the sense like nobody has done this in this scale. So basically, you take a blueprint of the information that you want to have on your mRNA, you code this into DNA plasmid, you put it in the bioreactor. The bioreactor actually, together with enzymes and nucleotides and so on, produces many copies of the spike protein encoded on the plasmid. Then you purify all of this and you get like pure mRNA with a spike and the right capping and stuff like that. This then you formulate, you put into this lipid nanoparticle stuff under pressure, you do some sterile filtration and you do aseptic fill and finish. And after 8 days, you basically have a batch of mRNA. So batch size at the moment is round about the runs in the back end here is like 80 grams. So basically 1 to 2 day LNP formulation gives you 1 million doses if you have enough raw materials. Then it has to sit for 3 weeks actually for like quality control testing and stuff like that, especially sterility testing. So this is the process. And now we try to debottleneck as much as possible by getting as many partners as possible in the last couple of months. So we have now, I left out the Pfizer sites for right now, but what we have, like, we have this big Marburg site with 1 billion capacity at the end of the year. We have our Mainz site where we produce messenger RNA drug substance, and we get a bunch of partners going online now. It's a very complex network of partners. So basically, none of these were existent. I mean, none of these were existent except Mainz last September. So Marburg just came in September, and all the others came online as well only during that time. So all of this is now, it's a mixture of like some do only fill and finish, some do drug product formulation, some do drug substance and so on. And this we have to manage. And on top of that, we have this whole Pfizer network. But because of this complex network that we build up, we are able to produce 3 billion doses this year. So this is now the manufacturing complexity. And now in the last 3-4 minutes, actually, let me quickly have an outlook of like into the other pipeline. So we talked about the structure of the company. We talked about why is it mRNA that worked against COVID. We talked about how to produce COVID. We talked about why it was so fast and so on, why we need partners. And this is really a quick outlook on like, this is the rest of the platform. So yes, there is mRNA, and we can do so-called fixed dose combination, fixed vac cancer immunotherapies or individualized immunotherapies. Basically, what you do there is you use mRNA to encode cancer antigens. So in the fixed vac, it's a set of very well-suited antigens, so 3 or 4 or 5 per cancer, that you can use a hard code, and then you can put it on the shelf. It's basically an off-the-shelf vaccine, cancer vaccine, because you prime the immune system with these antigens. And the iNeST is the individualized version of this. That basically means because cancer is different from patient to patient, and it's different by 95% actually, we can actually, we do sequence the biopsy of the tumor of the patient, and then we pick out the most significant or targetable mutations from that cancer. We encode these in mRNA, so up to 20 mutations in mRNA, and then we inject it to the body, and then the body actually learns to look for the cancer cells that have any of these mutated antigens. So this is so targeted, it's really a custom tailor-made vaccine for a cancer patient. So the immune system that doesn't see the cancer because the cancer is hiding, after presenting this mRNA, the immune system sees it again and can take out or kill the cancer cells that look exactly like the blueprint it was shown. The good thing is the immune system only goes wild against the cancer cells. So in that sense, it's almost a very healthy process. You don't have side effects. You have a fever because the immune system is activated, but you don't have hair loss. You don't have toxicity in the body. So it's a very well tolerated actually way of doing that. So that's mRNA for cancer vaccines. Then we have cell and gene therapies as well. So we have a lot of proprietary targets from 20 years of research. We have a big antigen library, and then we have CAR T technology against some of these proprietary targets. And with mRNA, you can, that's our novel invention actually, you can prolong the duration of the response in the body by using mRNA to amplify the cell and gene therapy, which is a new thing as well. So not only the targets that we're using are proprietary, but also the mRNA boosting is new. Then we have some, let's call it more classical antibodies, but with very finicky mechanisms. We have next generation immunomodulators together with Genmab, for example. We have like 2 bispecific antibodies in the clinical trials with very, very interesting, very targeted mechanisms. We do have even a small molecule immunomodulator in the program that you can then combine with other immuno-oncology agents. And we have engineered cytokines. So basically with mRNA, you can engineer cytokines or even antibodies, produce them in the body. So you don't have to produce them outside, but in the body. It's very effective, very cost effective as well, and very efficient in the body. And you can do many, many, many interesting things with that. So that's what we're working on. So now the question is, why do we have all of these broad platforms? Because every cancer is different. So you have cancers that are with a high mutational burden. You have cancers with low mutational burden. You have immune desert cancers. You have cancers with beta-2 microglobulin loss, so basically escape mechanisms. You have refractory tumors. And depending on what kind of cancer you have, you can use certain of these toolboxes. So basically, for example, for high mutational burden, mRNA neoantigen, so these individualized therapies are actually very good ones. So for low mutational burden, it's probably the shared antigens, for example, the FixVac or the CAR T cell or the boosted CAR T. For immune desert, it's probably RNA again, but also ribocytokines that I mentioned. Then there are CARs that are good for these loss mechanism cancers, and cell therapies and combination therapies for refractory tumors. So that's why we are, let's call it technology agnostic. That's why we have this like big broad pipeline of like technologies. So our competitor, whom we very value, actually Moderna, who has the other messenger RNA vaccine on the market basically as fast as we did it, and they have mRNA and they do a lot of stuff with mRNA. We say we have a lot of platforms, including the very mature mRNA platform, but we use it all of these various platforms to optimize the immune response and go after cancers and infectious diseases. So ours is actually technology agnostic, but focused on cancer and infectious diseases, whereas Moderna has like one technology, but they go broad in the therapeutic areas. Yeah, that's what they do. This is where we think you need this arsenal of like technologies to really make an impact on cancer. So now, very last slide, the outlook. So what are we going to do this year actually? So first of all, Comirnaty. So COVID vaccine needs to get to more than 1 billion people, ideally 1.5+ billion people worldwide. Then we want to do our oncology programs. We kept on doing them during the pandemic, but because of the patient recruitment was like slowed down a little bit, we have to refocus again and accelerate it. Want to bring 3 into randomized phase 2 trials. Some of those will have registrational potential actually as well. We want to extend our mRNA technology into new disease areas. We want to keep on updating our Comirnaty against COVID for new variants and so on. We have to expand our global capabilities, be it like marketing and sales, but especially footprint in the US, in Europe and Asia. We just announced that we're going to do a joint venture with Fosun in China, and that we want to build a big mRNA factory in Singapore. And we need to hire and hire and hire because we're growing so fast. So when I joined the company 7 years ago, it was 300 people living there or working there, and now it's 2,300 actually. So we gained 2,000, and this is just the beginning. If you look at what we're going to do. So long term, that means we want to usher into a new era of individualized cancer vaccines. We want to build a global business, commercialize our own products, and actually become a 21st century immunotherapy powerhouse. But first, we really have to control the pandemic, because otherwise the world will not really work again. So first and foremost, before we do all of that, we have to produce and produce and produce vaccine for the world. So with that, I'm through, and happy to take any comments and questions.
Okay, Dr. Poetting, thank you so much for your wonderful, fantastic presentation. You have mentioned Moderna. New York Magazine reported in December that Moderna's vaccine had been designed by January the 13th, just 2 days after the genetic sequence of the coronavirus was made public. The design of that vaccine took only 1 weekend, and by a month later, it had already been shipped to the NIH to begin its phase 1 clinical trial. Pfizer BioNTech vaccine received approval first. So was your design process as faster? And what was the role that artificial intelligence played in the development of your vaccine? Was it in early molecular development or at a later stage during clinical trial analysis?
A very good question. So we did start a little bit later, I think. So what did I say? So I think on end of January we had this kitchen discussion between Özlem and Ugur, and that's when we started. It is actually true that with mRNA, I mean, the target, the natural target to use in the vaccine was the spike protein. Yeah, but you could have used others. There was a ribosome binding site and stuff like that. But so we also said, like, it's probably the spike protein. So yes, picking that and encoding this into mRNA honestly really takes only a couple of days. So yes, we started in that sense, I think probably 2 weeks later, but a couple of days after we decided, we had also our first mRNA samples, 20 to 30 actually of those produced and having them in preclinical testing. Yeah. So what then happened, I think, Moderna, because they did more, while we focused on cancer vaccines, they had more experience in infectious disease vaccines, and they were allowed to go into the clinical trials a little bit faster. So we had discussions with our regulatory authorities in Germany, and this was the first time they saw this, and they were a little bit hesitant. So we had to convince them with extra data actually before we could go into clinical trials in April. So in that sense, we were almost as fast as with a construct, but we just couldn't get clinical data yet. But we used the time to get actually like 4 compounds into the clinical study. So we had like our various pieces of mRNA. So we used our immunoactive mRNA, the non-immunoactive mRNA, the self-amplification mRNA, and we used like the ribosome binding site as well as the spike protein. So we had like 4 in clinical trials then in April and May. And now comes the part with the data and the artificial intelligence. We evaluated all data and did predictions, and yes, we did use for binding simulation, stuff like that, artificial intelligence and so on. But we also used a lot of every piece of data that we could get from the clinical trials, and we decided on the candidate to go into phase 2-3 trial the day before we started the clinical trial. We did everything in parallel. We had like couple of candidates for selection, and I think two were likely, but we didn't know which one, and we produced clinical trial material for both, and we only picked the day before we ended the trial because we waited for the last bit of data and then swapped the candidate. So that has to do with, like, this is the power of data actually. We didn't do the large scale trial with the other compound, but probably that gave you a couple of more percentage points in the efficacy. So then, but in that sense, data is really everything, especially when you do that, but collect as much as possible. Last point on why we were actually first, I think because of having Pfizer as the execution machine on the clinical trials, that made us the first one because of this excellent process from our partner. To be honest, we brought the science, and Pfizer brought the scale up to get this really through the clinic, actually.
Thank you so much. mRNA and RNA was a technology that held huge promises for some time, but had run into biological roadblocks. How did you manage to make it work?
Yeah, It's also a very good question, because this goes back. I mean, this shows probably how we work as a company. So yes, mRNA has been used. One was like had big failures like 10 years ago, 12 years ago, and everybody basically gave up mRNA. Whereas our founders and Ugo Sain, he was always convinced that this is the way to go. But there were many steps that had to be optimized, and it took a while. I mean, it took honestly literally 20 years to optimize the last piece of it. The cap structures, how does it get into the body, how do you design the linkers between the data points on the mRNA and stuff like that. So it took really very tedious, tiny iteration steps actually to get this going and understanding what it does. I mean, whereas let's call it the conventional medicine, you have like a target and then you do a high throughput screening and see what sticks. This was more or less like a really driven or targeted development process to understand what's going on and find the best one. So I think with this like 20 years of experience, we were now in the position to fight the pandemic. It was the right time. We were lucky that everything was ready. And of course, we learned already. I mean, we had mRNA in the clinic since 2012 already. We learned, of course, from the cancer vaccines. So all of this together, learning from the clinical trials, from the cancer vaccines, learning from the data that we generated over 20 years, we were at this point that we were able to use it really in a targeted way.
And Doctor, we have heard that this is really a revolution, like no other in the development of vaccines. Are you already thinking about using this technology for other possible pathologies? You mentioned AIDS, but for example, Ebola, dengue fever, other?
Yes. So because the great thing about the mRNA is, if it works, you have the production process already figured out. So basically all the capacity that we are building right now, you can use for everything else. I mean, it's tiny differences, maybe use a different formulation or so, but in principle, the process is the same. So first of all, that's also why we are not so scared, let's call it, by the variants, because if there's a variant, we have the process already, and we can actually design a new mRNA very quickly and get this up and running. So that's one thing. We want to use it for other variants if necessary. Unless the virus mutates so badly that it doesn't work at all in principle. So the other thing is, of course, and now you can retackle infectious diseases that haven't been able to be tackled before. So yes, we want to look at AIDS. AIDS is very tricky. I mean, you have to. It's not. Let's call it as easy as COVID, because it kind of like mutates so quickly and you can grab it right away. But you can add mechanisms to the mRNA that potentially follow that. So in that sense, yes, we want to tackle HIV, we want to tackle tuberculosis. You could think about other kind of dengue fever, malaria and stuff like that. We have to evaluate what the mechanism is and then see whether mRNA is possible. But in principle, yes, the whole range of products needs to be looked at now. And some won't work, but hopefully many will.
Is it known that it is known that you're working on a new updated version of the COVID-19 vaccine? How different is it from the current one? And will it totally replace your current version, or will it be necessary a third dose after 2 doses of the one we know now?
Yeah, so I think that's 2 questions. And one is the extra doses. So initial data now suggests that you lose some immune responses. I mean, we couldn't follow that many data points yet because we only have a couple of months since we have like fully vaccinated people from the trial, but you see that the immune response is going down. And so I think what we publicly said is like we anticipate like probably after a year or so, you would probably need a booster shot, round about a year or so. The question then is, and that's what we are evaluating in clinical trials right now, how big is that boost then? I mean, it could be that the boost is then so high that you don't need another boost for 2-3 years or something like that. But maybe it's better to refresh it every year. So that we don't know yet. But we are preparing for booster shots. The second question is the variants. Of course, we look at all the variants. So we basically reproduce the variants in the laboratory. We use them on antibodies that we have, and we see how potent is this. And so far, all of the variants that we see they're still working. Some are working better than others, of course, or the vaccine is working better than some are working against others better than others. However, so far there seems to be a very good protection still. So far we didn't, or there is no new vaccine that we want to produce and get into the vaccination. I think for that you need a process, a worldwide process, where an agency or a conglomerate of agencies or network of agencies actually says like, okay, now this is a variant that we really need to do something against it, maybe in a local geographic area, maybe worldwide or so, and the scientific community decides on like, now we need a new one, and then we could easily tackle that. The question is then, do you combine it with a wild type? Do you give like an extra shot? We don't know yet, but that's all in part of studies that we're doing right now, preclinical and starting to do clinical.
And I have another question. In case of a third or fourth COVID-19 vaccine administered to the same individuals, how will you overcome the difficult issue of RNA immunogenicity? Intramuscular RNA is highly immunogenic per se in any form.
Okay, actually, honestly, on this question, I honestly have to pass because I'm not the expert on this one. So I'm just a physicist and do the operations. So that is a question that needs to go to our founders actually and the scientific personnel. So I honestly don't know.
And let me ask you to finish, how did a physicist make a career in the pharma industry? Curiosity, I think. So eventually, I think what I liked about physics is that you learn thinking, you learn to deal with complex problems. You have to learn how to structure complex problems, and I think that's how eventually I ended up in finance, because finance has to do with numbers and has to do with like complex problems. And I think it's great to have like these challenges, and you kind of like build this into a framework, into a decision framework. So you can really move the needle because you create clarity, and then you can go left or right and stuff like that. And that's what I really enjoyed, coming from theoretical physics to real operations. So consulting was a little bit theoretical to a certain extent, but then in the real world business or so, you could really implement things. And this was one thing at Novartis, of course, where I learned my toolbox. But now at BioNTech, I can really leverage all of that. And we have many complex problems, and it's great to be creative and tackle those. So I think there's this finance piece in me, and there's a physicist in me. That's why building a production network is probably exactly what I was meant to do. Okay, thank you so much. I think there are some questions from the public.
Thank you so much, Doctor. Thank you, Nora. It's been a real treat to have you. We are very proud to have you with us today, and we really want to thank you for your time. I think the only question I got from the public is something you spoke about recently, and the question was, what would happen if a person got 2 or more, 3 different vaccines, if this was positive, if this was good, and it would obtain a better result?
Yeah, so I think what initial studies, I mean, we are doing these studies as well. What I read about and what first data is about to come out is, for example, you get an AstraZeneca shot first and then a BioNTech vaccine. So it looks like that gets you a very good efficacy around about as if you had like a pure double BioNTech Pfizer vaccine. So so it means it's eventually looks like it's a higher than a pure AstraZeneca efficiency. However, so I haven't seen the data yet. So I think it's like a pre-shadowing of data that's about to come out. And I think scientists have to comment on this, but it looks that you can indeed combine vaccines and they have an effect. Yeah. It's a little bit like with a 2-shot. I mean, we have the 2-shot regimen, and a 1-shot gives you a certain protection already, but 2 are better because that's where we have the data, and we know that it's 95%. But I think that there are countries, and it was a good choice by many countries to start vaccinating people for the first time and extending the time to the second shot a little bit. so that you have like higher immunity in the population actually, and then start vaccinating with the second one. So I think in a normal world you would have done like 2 shots with a difference of like 3 weeks exactly. But I think in this messy pandemic situation, it was a very good choice to really do like many first shots and then follow up with the second ones.
So may I, Ivan, may I add one last question?
I was going to ask you, you had one last question.
Okay, thank you very much. Doctor, regarding cancer, are you developing other immunotherapeutic approaches such as galectins, for example, which show promise, a great promise in cancer treatment?
So, sorry, what was the subject?
Galectins, you know, the proteins and sugar.
Yeah. Okay. So I think it's the great thing about the mRNA vaccines is that again, once you have the structure worked out, you can go against many cancers. So 2 examples. I did this. I told you about this FixVac program. So with that, if you have the right mix of antigens. So for example, there's a HER2 antigen on breast cancer, and there are others for prostate cancer and so on. So, but each cancer has probably like the perfect set of antigens. And it used to be very difficult to have like a vaccine against many of these antigens. It was only one antigen you could target, and that worked like so. Sometimes it worked, sometimes it didn't. Sometimes it only worked in certain circumstances and so on. But now it's very easy because you can just add 1, 2, 3, 4, 5 of these antigens into one vial and just apply the vaccine, the cancer vaccine, with one shot. And then the immune system is actually looking for all of these antigens at the same time. So I think that's great, because that means you can combine for each cancer type the perfect set of antigens. And having this, the framework, you can do 3 on breast cancer, 2 on prostate cancer, 6 on skin cancer or something like that. It's always the same structure. You just have to pick the antigens and the rest is there. It's the same with like the individualized mRNA, where we actually pick the 20 antigens for each patient anyway at the same time, for each time. So 20 different ones for each patient, and the production process is always the same. So I mean, you could think about it like a machine that can do any medicine. And I think we are not that far from that. So we are in clinical trials with those things, and I think this will be a totally new world. The only thing is we need more and more data to really prove that it works.
Okay, thank you so much.
You're welcome.
Thank you so much, Doctor. Congratulations on your good work, and thank you so much for giving us this exclusive interview and for your time. It's been a pleasure to have you. Nora, muchísimas gracias. Thank you so much. I enjoyed your interview so much, and we really appreciate it. It's been a pleasure to have you.
Thank you so much. Thanks for the invitation. It was a pleasure to be here. And thank you, Nora. Thank you, Ivan. Thank you.
Thank you, Doctor. Muchísimas gracias. Un honor.
Good evening to both of you. Bueno, muchísimas gracias a los dos. Ha sido un lujo contar con estas dos eminencias y poder ser partícipe de esta entrevista.